Hello everyone, myself Naveen Kumar Naidu. Today I am going to talk about EGFR targeted therapy focusing on GEFITINIB. So first, EGFR stands for epidermal growth factor receptor. It's a protein that sits on the surface of our cells and acts like a switch for growth and division. In many cancers, this switch is either stuck in the position of because of mutations or it's overproduced that drives uncontrolled cell growth, essentially cancer. EGFR is detected by some techniques that are IHC, immunohistochemistry, checks protein levels in tissue samples, FISH, fluorescence in situ hybridization. It looks for gene amplification and lost and more advanced methods like PCR or NGS. PCR stands for polymerase chain reaction and NGS stands for next generation sequencing. These are helps to check for specific mutations in the DNA itself. Treatment of options for patients with EGFR mutations. Traditionally, chemotherapy kills both healthy and cancer cells, so patients often suffer severe side effects. With precision medicine, we can target EGFR more specifically. Antibodies like cetuximab block the receptor from the outside. Small molecules like gefitinib enter the cell and block the receptor's internal enzyme, which is kinase enzyme. Patients with EGFR mutant tumors usually respond much better to these targeted drugs compared to chemotherapy. Discovery of gefitinib. In the 1990s, the gefitinib is developed by the scientist AstraZeneca by screening of thousands of chemical compounds, basically testing chemical compounds to see which ones could shut down EGFR's kinase activity. They found a quinazoline based molecule optimized it so it fit exactly into EGFR's ATP binding pocket and that became gefitinib. The mechanism of gefitinib is gefitinib blocks the ATP site. EGFR tyrosine kinase inhibitor. The blocking of ATP without ATP, EGFR cannot send growth signals to move, and then automatically the cancer cells division will be stopped. Evidence of efficacy. In lab experiments, gefitinib slowed the growth of colon, breast, and ovarian cancer cells that carried active EGFR. In mouse models with human tumors, gefitinib delayed tumor growth significantly, and when combined with chemotherapy drug Paslic, the effect of was even stronger. The tumors shrank more, and mice lived longer when combined with chemotherapy drugs, etc. Precision medicine impact. The key to success with gefitinib is patient selection. Not every cancer patient will respond. Only those whose tumors have activating EGFR mutations. So today, genetic testing is used before prescribing gefitinib. This means the benefits and approaches are patients without the mutation avoid ineffective treatment. Patients with the mutation have a much higher chance of response. Overall, this improves survival, reduces side effects, and saves healthcare cost. This is a great example of how precision medicine has transformed cancer treatment. Finally, the conclusion of this gefitinib is gefitinib was the first targeted EGFR inhibitor to reach patients. EGFR is a critical growth receptor and its mutations drive cancer. Gefitinib was developed through screening and designed to block EGFR activity. Preclinical studies showed stronger efficacy, especially when combined with chemotherapy. Most importantly, precision medicine matching the right drug to the right patient has made treatment more effective and safer. Thank you.